Today, I'm going to present some of the work my lab's been doing over the last several years to try to develop an animal model of SDHB-related pheochromocytomas. I should point out that my name is Justin Annis. I'm at Stanford University, and I clinically focus on hereditary neuroendocrine tumors, specifically the hereditary pheochromocytoma and paraganglioma. And I have no financial disclosures, but I also participate in the medical advisory board for the Pheopara Alliance. A really central goal of my lab is to develop an effective treatment for SDHX-related tumors and cancer. So when I say SDHX, that's because there's a variety of different forms of SDH mutations. SDH stands for succinate dehydrogenase. There's A, B, C, and D. Specifically, the work today will focus on SDHB, but it's applicable, we hope, to the other forms as well. So in order to develop a treatment or an effective treatment for SDH cancer and tumors, we really need to better understand how these tumors form. So that's first and foremost. And you can't do that easily in people because you can't watch the whole process of tumor formation. You can only collect the tumor at the end, possibly from surgery, but you don't know how it began and how you got there. Also, you need a way to test new drugs and new therapeutics. And human tumors are very, very hard to grow outside the body, and they lose a lot of the relevant features. And then inside people, of course, we can't just test chemicals to see what happens. So my laboratory, what I thought was what we really need is an animal model, a mouse model that really is very similar to the human disease, which will form these tumors. And then we can use that mouse model to learn about how SDH tumors form and then to identify therapeutics. Okay. So in order to understand what my lab has tried to do with making a mouse model of SDH, I think it's important to take a step back and understand how do SDH tumors form, SDH-related tumors. So every cell in your body has DNA, chromosomes, and you have 46 chromosomes. You got half of them from your mom and half of them from the dad. In a person who doesn't have an SDHB or SDHG mutation, they have two copies of the SDHB gene. Both of them are the wild type or normal sequence. Individuals who inherit a mutation in the SDHB gene now have one copy that's disrupted and it doesn't work, and the other copy they have is totally normal. And this sometimes is referred to as a carrier state. Now, all your cells in your body, if you've inherited an SDHB gene mutation, have one broken copy and one normal copy. And this is 100% sufficient for everyday function, normal function. The cells do everything just fine. And you can do sports, you can exercise, fully normal. But then rarely in some cell types, if you lose the other copy of SDHB, now you have two broken copies, then that can initiate tumor formation. And so many people think about it if they were to imagine what happens is that they have an SDHB tumor and then the other copy breaks when a cell divides. This is in fact not the case. What actually happens is that you're born with a broken copy of SDHB. And then in rare events during cell division, a mistake is made in the cells that can form paragangliomas or pheochromocytoma. And you actually lose a substantial chunk of the DNA in that cell, including SDHB, which leads to the loss of what we think is about hundreds of genes. Okay, so when you think about SDHB tumors, they don't have any working copy of SDHB, but they've also lost a large chunk of DNA. And so this is kind of how you should think about how do you model this in an animal. And one of the we wanted to start with was the simplest question, which is SDHB deficiency? So if you only lose, if you do this first model, you only lose SDHB, is that sufficient for tumor formation? So currently, the way the field has been thinking about SDH-related tumorigenesis is that we've noticed that if you have mutations in any of these group of genes that are involved in ATP generation in the mitochondria, they all result in a spectrum of tumor formation. And the prevailing idea is that, let's imagine you had a broken copy, you lost both your copies of SDH, succinate dehydrogenase. The function of the succinate dehydrogenase enzyme is to convert succinate to fumarate. If SDH is broken, you get a very large accumulation of succinate. And the idea is that this metabolite, succinate, is cancer promoting because it reprograms the cell in a variety of detailed molecular ways that I'm not going into. But the central idea of SDH biology right now is that you get succinate accumulation, and that accumulation causes cellular reprogramming and tumor formation. And this is the idea that we were testing in our model and ultimately proved is not really sufficient to explain why cells get tumors. Or sorry, why SDHB deficiency itself is not sufficient for forming tumors. Okay, and I should point out, we're not the first people to try to make an SDH model. In fact, this has been going on for about 20 years or so. And we've learned from what others have tried. And so we took different approaches. And one challenge to us was at the beginning when we started, it wasn't even clear that we would be able to do this at all. In fact, some had concluded that it's basically impossible for SDHB deficient cells to survive in the mouse. Okay, so the trick that we did, or one of the important things we did, was we only deleted SDH or removed SDH protein in the cells that form pheochromocytomas and paragangliomas. That way we wouldn't worry about causing damage to other tissues. Also, in the cells that we removed SDHB, we labeled them green. So they turned on a fluorescent protein so that we can specifically know which cells didn't have SDHB. And when we did this, and we deleted both copies of SDHB, so here you're looking actually at a mouse adrenal gland. This outside is the cortex area. This inside is the medulla. These are the cells that give rise to pheochromocytomas. So when you delete, and all of these green cells don't have SDHB. And when you do that, you do not get any tumor formation. The cells survive just fine. They're not increased growth, they're not decreased growth. They totally survive, but there's no tumor formation. And so this was our first insight that SDHB deletion or loss of SDHB is not sufficient for tumor regeneration. The hypothesis is that very high levels of succinate are needed to drive tumor formation. And so we use a very advanced technology to measure succinate levels in our tumors. And here, the brighter the red, the more succinate. You can see here, the samples from our SDHB deficient mice are very, very succinate rich compared to normal cells. So we have no tumor formation, but we have massive levels of succinate, saying that this old hypothesis that succinate is sufficient for tumorigenesis or tumor formation is wrong. But importantly, this is looking at electron micrograph as a very, very high resolution picture of SDHB deficient cells. And what they show, these structures here are the mitochondria. These are where cells generate ATP. These guys are normal. In our SDHB deficient cells, you have massive expansion of these. They're very large. And in fact, this is exactly what we see in the SDHB deficient tumors. So when you lose SDHB, you do get this very important metabolic reprogramming, but you don't get tumor formation. Very high levels of succinate, but no tumor formation. And so we hypothesized that what was missing was something to push the cells into cycle, to make the cells grow. If the cells aren't forced to grow, at least to initiate, then they won't form tumors. And so SDHB deletion alone is not enough. We have to add something else to push the cells to grow. And we used this gene called neurofibromatosis, which on its own can also cause pheochromocytomas. And this caused an increase, in the growth of cells. And then as you can see here in the mouse, it caused tumor formation. So when you have SDHB deletion alone, the medulla, you don't get pheochromocytomas, but when you delete them both, both succinate dehydrogenase B and neurofibromatosis, you get tumor formation. And there was high succinate there. So the bottom line from our work, I think the most important principle that we identified is that when you have a single copy of SDHB that's deficient, that's missing, cells are totally normal. If you just delete the backup copy of SDHB alone, you don't get tumor formation. We think that there's additional genes that are deleted at the same time that allow tumor formation to happen. And currently, we don't know exactly what those additional genes are. We used neurofibromatosis 1, the NF1 gene, but that's probably not what happens in most people. Okay, so then the last part, important question is, okay, now that you have this new mouse model, what's next and what did you exactly learn? So this is a picture of succinate dehydrogenase, the enzyme, and it's made up of 4 different subunits, A, B, C, and D. And if you have a mutation in any of these, you're predisposed to getting pheochromocytomas and paragangliomas. But what you'll know is that people with SDHB mutations have different risks than people with SDHD gene mutations. And one question that's been difficult to understand is, why is this the case? And so why do different SDH gene mutations behave differently? And our explanation is that it's probably because when you get the loss of that chromosomal chunk, there's different genes that are lost because SDHA, B, C, and D are all located in different parts of the genome. And so it's those other genes that are controlling why SDHA mutations are different than SDHB mutations. And so we can use our mouse model to try to uncover what are those important genes that are being deleted that are driving tumor formation. And then really importantly, we can use our mouse model to identify new treatments, new drugs by treating the mice, and then use genetics to identify new drug targets. Okay, and then I just want to take a moment to identify Neely Armstrong in my lab, who did almost all of this work, or she directed a lot of it, and it's taken 6 or 7 years for her to do it. So it was a big effort.
We extend our greatest appreciation and excitement about these findings, and please convey this information to your whole team and to Neely and to everybody who's worked on this. It's really, really exciting.
Well, thank you very much. I think, yeah, like I said, it's been a number of years, and as many of us that the Alliance membership know, this is a tough field to do science. It's a tough field to be a patient in, because it's resource limited. And so everything that people do to participate at any level, whether it's using your voice or using your wallet or just learning, I think it's great to be a part of this community and to keep forwarding the effort to make sure that we get the resources we need.